Hello and welcome back to part 4 of enveloped negative strand RNA virus with segmented RNA as their genome. I'm Dr. Elijah. So previous lectures we've talked about the family of Octomyxoviridae as well as some example of the viruses that fall into the member of the family of Octomyxoviridae. And we also look at some of the characteristics of flu A, flu B, and flu C, and what are the differences and similarity between the viruses. Then we also look at the detailed structure of the virus, Influenza A virus. And today what we're going to look at is the life cycle of this Influenza A virus. So the diagrams here show an overview of how the influenza virus attach to the receptors and then undergo endocytosis and then release their the genome of virus into it, and then they started to produce mRNA, and then the mRNA will be used to produce the protein, translated to produce the protein, where the protein will then go back into the nucleus, and then once the viral protein is sufficient, then they move on to the fourth step, which is genome replications, produce a lot of viral genome, and then they assemble at the cytoplasm and eventually bud out from the cell. So this is basically the overview, and what we're going to look at is the detail every single step and how the viral protein is being produced as well as the genome replications, and in the end produce a new viral progeny. So let's start by looking at the viral protein, glycoprotein, that display on the surface of the virus. And we know that for flu virus, there are two glycoproteins that is being displayed on the surface, which is HA and NA. But the one that is responsible for the binding of the virus and also fusions of virus into the host cell is the HA, or heme agglutinin protein. So heme agglutinin protein is, like I mentioned earlier, is a homotrimers. So the size is about 110 kilodaltons. And during the productions of the translation of this protein, it has signal peptides which fall on here that signal the protein to go to ER to be processed. And what we can see from here, this is the HN protein, where that is the N terminal and over here is the C terminal. And what we can see here, there's a fusion peptides that located here. And arrow here shows that the site where the protein will be cleaved by cellular trypsin-like proteinase, meaning that this protein will be cleaved into two portions, one here and the other one is here. So over here is HA1 and over here is HA2, where HA2 has the fusion peptide. Alright? Now, bear in mind, even though the protein has been cleaved into two, the protein are still linked together by here, show here, disulfide bond. So this disulfide bond that link the HA2 and HA1 together. So the HA protein are still intact, and just that they have been separated into two. Alright? So underneath the red line or the protein, what we can see here is these are the symbol of all the or carbohydrate that are being added onto the protein. That's why it's called glycoprotein. Now this glycoprotein is quite important in the sense that not only it makes the protein become glycoprotein, it also gives the virus to be the pathogenicity of the virus. Alright, so now that we know that HA has a fusion peptide, then we know that, okay, not only involved in binding, and it also involved in the fusion of the virus. So listed here are some of the explanations of how HA mediated the membrane fusions. So you can look at this slide as I explain using the next slide. Over here, what we can see is the virus have HA protein that display on the surface of the virus, and the receptors that on the host cell that will be bound by the HA protein is sialic acid receptors. This is exactly the same as the Newcastle disease virus. Right, later on we'll look at there are two forms of sialic acid, whether they are alpha 2,3 or alpha 2,6. We'll leave it till later. But right now it's suffice for us to know that the HA will bind to sialic acid receptor on the surface of the host cell. So once it's bound, the cell will then undergo endocytosis into the cytoplasm. And we know that when the endosome enters the cytoplasm, in the cytoplasm, the pH of the endosome will start to decrease to become acidic. So when this happens, it causes the conformational changes of the HM protein. Remember the HA protein. Remember the HA protein is being cleaved into two, where the fusion peptide is at HA2. So, but because in normal condition, the fusion peptide is being protected by the HA1. Alright, this fusion peptide is being protected by HA1. They are being covered so that the virus will not simply fuse into any cell or membrane that they come across. So when these conformational changes happen during the decrease of the pH, alright, the acid, right, it becomes acidic. So it causes the conformational change of the HA protein. So as shown here, the fusion peptide is being protected, covered. So once the HA protein, the pH changes the HA, it causes the fusion peptide to be exposed. And what happened here? The fusion peptide will now hook into the membrane of the endosome. So like the previous explanation of Newcastle disease virus, and this alpha helix, the helix bundle, will now try to pull the two membrane closer to each other, and eventually they fuse. Alright, so in Newcastle disease virus, it's quite straightforward. Once it's fused, the ribonucleoprotein or the MPPL with RNA will then be released into the cytoplasm of the host cell. But for influenza virus, it is slightly different. Even though the membrane has been fused, the ribonucleoprotein will not be released yet. And what happened here is the ribonucleoprotein of the influenza virus is still attached with the matrix protein inside the core of the virus. So what happened next is, remember there is ion channel here. So this is the ribonucleoprotein, is still attached to the matrix protein underneath the membrane. So even though the membrane has been fused, but the ribonucleoprotein is still attached to the matrix. So matrix protein. So here we have this ion channel. This is the one that is responsible for pumping the proton or the acid into the core. So back here, what we can see is the core of the virus is still remain the same as the original, and when the fusions happen, the ion channel started, the M2 started to pump the proton into the core of the virus. So once this happened, the acid will causes the conformational changes inside the core of the virus that resulted in the RNP being released into the cytoplasm. Alright, so it's more of like two steps. One step is the virus undergo endocytosis, pH changes, causes the conformational change of the HM protein. It resulted in the fusion of the two membrane, and the ribonucleoprotein will not release yet until the core of the viral protein, the core of the virus has been reduced to acidic pH. Only then the ribonucleoprotein will dissociate itself from matrix protein and then eventually release into the cytoplasm of the host. Alright, so moving on to the next steps. Now, we have done with the first step, which is attachment, then fusion. The next step will be synthesis of the viral proteins. So like NDV, we learned that NDV immediately use their ribonucleoprotein, which is the polymerase that associate with the MP and the phosphoprotein. Then they'll start synthesizing the mRNA. However, for influenza virus, it is not as straightforward as NDV, because NDV polymerase can synthesize the mRNA, which then cap the mRNA and synthesize a poly-A tail. So the polymerase for NDV has these additional functions where they synthesize the cap. However, for influenza virus, the polymerase does not have capping functions. Alright? So when they do not have capping functions, where can they get the cap? So the only way that they can get the cap is in the nucleus where there are a lot of mRNA. So the virus have gone through all these evolutions and making sure that their ribonucleoprotein will now be translocated into the nucleus where there are a lot of the cap. Alright, a lot of the mRNA that have caps. So on the ribonucleoprotein or RNP, okay, this is the shape of the RNP. The RNA is here, which is protected by MP protein. Alright, protected by MP protein. And the protein, this is a simple version. Alright, RNA is in the center where it's protected by MP. And at the end, you can see there are PA, PB1, and PB2, which is the polymerase that binds to the 3' end and 5' end of the RNA that make it like a frying pan. Okay, we call it this as a pan-handle structure, just like pan where you hold this like a pan. Okay, so this is the 3D structure of the ribonucleoprotein. So you have PA, PB1, and PB2, and RNA with the MP. Now, unlike NDV, this influenza virus doesn't require any rule of 6 for the MP to bind to. Alright? Okay, now what happened next is the ribonucleoprotein, okay, will now be translocated into the nucleus. Now, how does this happen? The ribonucleoprotein has nuclear localization signal. Okay, this nuclear localization signal, or in short, is PKK. Sorry, this is supposed to be here. PKKK. All right, proline, lysine, arginine, lysine, and valine. So this PKK KV is a nuclear localization signals, which will now bound by cellular protein, importin alpha, and lead to the recruitment of importin beta. So these two proteins are the cellular protein that are responsible to translocate the viral nucleoprotein into the nucleus. Right? So these are the two proteins that facilitates the transport of the RNP through the nuclear pore complex into the nucleus. So now the 8 segments of the influenza virus RNP is now being transported into the nucleus. So once in the nucleus, transcription can happen. Alright, so I would like to introduce you to the influenza A promoters. So for Newcastle disease virus, we know that the 3' end has a leader sequence, then you have MPP, MF, HNL, and the trailer. So I mentioned to you earlier on that the 3' end is the site where the polymerase binds to, and they are the promoters. So for influenza virus, as we see here, the RNA is already bound by the ribonucleoprotein, the PA, PB1, PB2. So the protein, the RNA is already bound to the promoter site. But let's look at where exactly is the promoter for the influenza virus. So this is the pan-handle structures. Alright. So over here we notice that there are 3 pairs of nucleotides. So 5' end and 3' end. It's a negative strand RNA. So the 3' end and 5' end is actually quite complement to each other, and there are many of the nucleotides are quite complement. But what we notice is that there are 3 nucleotides that is really important in this case. AGG here, UCC. These are the 3 nucleotides that is very important for the virus replication. So we call it wild type. WT stands for wild type. In wild type, when we look at the sequence, it's AGG and UCC. So scientists started to wondering, are these really that important? So what they do is, when you use wild type sequence, what you produce is 7 times 10 to the power 8 PFU. Okay, but when they start to do mutations on the A, so in the end you produce the G and the C complement, right? They notice that there are twofold reduce of the virus that being produced when this mutation happen. 7 times 10 to the power 8 become 3 times 10 to the power 8. So the next mutation they did is changing the G to U, and what happened here? There are 4-fold reductions of the viral proteins that being produced, 1 times 10 to the power 4, showing that. And then the next one is they mutate this one, A. So you have 1-fold reductions, 1 log reductions of this, 4 log reductions from 8 to 10 to the power 4, from 8 to 10 to the power 7. So they shows that these positions of the 3 nucleotide is extremely important for the virus replications, right? So this is basically just to show you where the promoters sequence on where the promoters is located, and very important for the virus replication. So, like I mentioned earlier on, that the polymerase of influenza virus doesn't have a capping functions. That's why they need to find the cap, alright? They find the cap. So in this case, because they cannot produce the cap, what they can do is they carry out a process called cap snatching. Snatch, the thief snatch handbag or whatever. So the polymerase here, they snatch the mRNA that contained cap from the host mRNA. So the viral RDRP, RNA-dependent RNA polymerase, doesn't have a capping activity. So they have to carry out a cap snatching, to steal the cap from the cellular mRNA. Let's move on to, this is the common conditions in the nucleus, where we have DNA that undergo transcriptions. You produce the exon, intron, exon, intron, exon. And then the next step, this is the pre-mRNA, and this is also pre-mRNA. The next step is adding a cap to this pre-mRNA, then undergo excisions of intron to remove the intron and splice the exon, join them together, splicing, joining the exon together, and add the poly A tail. Eventually, this is the complete mature mRNA. So I hope you still remember the characteristic of mRNA. They must have cap, they must undergo splicing, and they have poly A tail. So in the nucleus, we know that this is the site where there are a lot of mRNA or pre-mRNA that being produced inside the nucleus. That's the reason why influenza doesn't stay in the cytoplasm. The RNP doesn't stay in the cytoplasm, wait for the mRNA to come out. Instead, they go into the nucleus, where the nucleus has abundance of the mRNA that being produced constantly. So what they do is, having this RNP, PA, PB1, and PB2, this is where the polymerase snatch the mRNA that has cap from the host. Now, I know this is slightly complicated, but please bear with me. So we mentioned that the RNP has, or RNA-dependent RNA polymerase, has 3 units, PA, PB1, and PB2. Each of these has their functions. Alright. First of all, look at PB2. So PB2 has a domain where it's called cap binding domain. So this domain on the PB2 is the one that's responsible to identify mRNA that has cap and then bind to it. Alright, this is where mRNA cap binds to. And you notice that the distance between PA and PB2 is about 50 Armstrong. So what happens next is when the mRNA is bound, the cap is bound to PB2, because the distance between these two is 50 Armstrong, is almost 10 to 13 nucleotide distance. So the mRNA come to the PA where there is a domain for endonuclease domain. So the nucleotide, the original mRNA nucleotide, when they reach here, the endonuclease will now cleave the mRNA about 10 to 13 nucleotide from the cap. So now we have cap with short mRNA 10 to 13 nucleotide, which is originally from the host. And what happened next is it has served its purpose where it binds to the cap. It has served this PA has served its purpose where it has the endonuclease to cleave the nucleotide. So now this nucleotide from the host is being put into PB1. So look at here, PB1 have 4 channels. One is called template acid, product acid, NTP entrance, as well as the channel where the RNA of the virus is located. Okay, so in the center is where the PB1 active site is located. So now we have the mRNA from the host that go into the product acid. Okay, that go into the product acid. And now what we see here is the A and G will now complement with the U and C of the viral genome. So now we have 2 complement. Okay, A and G from the host mRNA. This is the 3' end of the viral genome, where it has U and C nucleotide. Okay, now complement and complement, good. So we now have this serve as a primer for the PB1, which serve as the polymerase to start adding nucleotide. So what happened here is NTP, you have AUCG nucleotide that comes in here. So now the next template is G. So nucleotide C come in, cytosine go in, then this template will now going out these directions, and the product will continue to go out. So nucleotide go in and go in and go in and go in. So eventually we are synthesizing an mRNA that has a cap, 10 to 13 nucleotide that come from the host, and the rest of this come from the virus. So this is the virus RNA that going up from the template acid. Right? As this go up, nucleotide keep adding in, and this keep going up. So this will become mRNA. This will be still the viral genome. Alright? So bear in mind that the 5' end of the RNA is still bound within PB1, which is on the 5' binding pocket. So this one go out, and the RNA still go out and go up, and eventually it come to a point where the RNA can no longer go out because the binding pocket will bound to it, and it come back. Alright, so this is how the mRNA is being initiated and synthesized and become polymerized. Right, so in NDV, towards the end of one gene, we call it gene N, there is 6 U that allow the polymerase to undergo polymerase stuttering and produce 120 poly A tail. what about influenza virus? And we can see from here the front end, okay, this is 3' end, the front end of the viral genome doesn't have U. Alright? But when we carefully analyze the genome of the virus, we notice that there is a stretch of poly-U in the middle of the genome. So this is the 5' end that go into the pocket. But before the 5' end, there is a stretch of poly-U right in the middle. So when polymerase started to synthesize, and they come to this U, they undergo the same process as NDV, undergo RNA polymerase stuttering, and it start synthesizing the poly-A tail here. So what we can see here is this stretch of RNA from the genome will not be part of the mRNA. Wherever that the U is located, that is the end of the mRNA. So you can see here you have AAAA, RNA polymerase stuttering that occur here. And eventually, what we produce is the mRNA that has 10 to 13 nucleotides that come from the host with the cap. Then viral genome, see, it starts with A and G that complement, alright, to the U and C from the host, from the viral genome, and then it continue with the rest of the viral genome. And then now we produce the mRNA. So this is the process that explains how the mRNA of the influenza virus that being produced. Alright? Okay, so we remember we have a total of 8 segments of RNA for influenza virus. So using this synthesis, we produce 8 mRNA. However, do you remember that I mentioned to you there are a total of 18 proteins that being produced by the 8 segments. So the mRNA that being produced by influenza A, they also undergo a process called splicing, right? Using the host spliceosome, where they splice the mRNA. For example, in this case, we have the we have this mRNA that being produced for M1. Alright, they're being produced for M1. The mRNA can be spliced where they remove the middle part and join the first part and the last part of the mRNA together to produce another protein which is called M2. So meaning that by using splicing techniques, the virus can again produce another 5 mRNA. So what we can see here is this is the full length, and full length of the new RNA segment that will produce mRNA. Then 5 mRNA, additional mRNA will be produced by using splicing in the nucleus. So we have PB2 that undergoes splicing to remove the middle part and joining these two. And now we have an additional mRNA called PB2S1. The same go for segment 7 and segment 8. So in segment 7, M1 will splice and produce M2, splice, produce M42. Right. 4 simply means that they are mRNA. There is another mRNA. Yeah, M1 actually produce 4 mRNA. So mRNA 1, mRNA 2, 3, and 4. But the mRNA 3 is still in the process of investigations, and scientists still couldn't find the protein that produced by the mRNA 3. Right. So then they found mRNA 4. That's why it's mRNA 1, mRNA 2, M42. Right. So 3 mRNA is being produced by segment 7 and 3 produced by segment A. So we have NS1, NS2 or NEP, nuclear export protein, and NS3. Alright, so original 8 segment produce 8 mRNA, then using splicing we produce an additional of 5 mRNA. So in total we already produced 13 mRNA. Alright, so bear in mind we say that the 8 segment produce a total of 18 protein. So there are 2 additional processes that carry out which is not involved inside the nucleus but later in the cytoplasm. We'll look at this later. Alright, so this step is called the ribosome alternative initiations. So the 13 mRNA produced in the nucleus will now be exported into the cytoplasm. So once in the cytoplasm, now the standard translations process will be carried out. I hope you still remember the the regulations of eukaryotic translation process. So you have cap, you have poly-A tail, so that will be quite standard. Then you have the EIF13, then EIF2, then bind to ribosome 43S pre-initiations complex. Then you have EIF4 that bind the mRNA and bring it together. So using that 13 mRNA, the ribosome will bind to it and start synthesizing the protein for the 13 mRNA. So what happened here is during the translations, alright, I hope you still remember when EIF4E binds to the cap and then bring it to the 43S pre-initiation complex, that will become 43S initiation complex dash mRNA. So this complex will then move along the RNA to find AUG. Once they found AUG, that is where EIF5 and 5B cleave the GTP to GTP and dissociate the whole EIF, and 60S ribosome binds to it and start synthesizing the adding the amino acid into it. Alright, so this is the standard method, but also there is alternative initiations. So during this scanning, E43S pre-initiation complex miss the first AUG, but rather go to the second AUG. So what happened here when they see the second AUG, the same process go on and dissociate all the EIF and ribosome binds to it, and this is where the first amino acid will be located compared to the original where it is synthesized from the first AUG. So what's the difference between this one and this one? In the end, what you're going to have is they might have the same C terminal when the reading frame is the same. They might have the same C terminal, but their N terminal is definitely different. Okay, so if we look back here, alternative initiations, here 1, 2, 3, 4. So what we can see from here is this is the original PB1. It starts from the first AUG, then N40 missed the first AUG. Instead of first AUG, it goes to the second AUG. So what we can see here is they have the same C terminal, but different N terminal. Alright, different N terminal. Like I mentioned earlier, that the ORF can be at the same reading frame or they can be at the different reading frame. And over here, what we can see here is that there's this PB1F2, they use a different reading frame, right? So here, this particular protein has no similarity or identical amino acid sequence compared to PB1 or N40. Whereas for here, PA, we have PA-N182 and PA-N155. So they all share the same C terminal, but the N terminal is different because they use the same reading frame but different AUG within the same reading frame. Alright, so with this, we already produce 1, 2, 3, 4, additional 4 protein. So 13 mRNA produce 13 protein plus 4 by using alternative initiations, we produce additional of 4 protein. And next, there's another one here, which is PAX. This protein is being produced by a process called ribosomal frame shift. So what does it mean by ribosomal frame shift? Over here, this is the original sequence. It starts with AUG. It's synthesized, and we know the anticodon will recognize the codon. For example, in this case, serine has the sequence of UCC. The anticodon will be A to U, G to C, G to C. Alright? Then the next one is UUU. So the one supposed to go in is AAA, but instead the anticodon that goes in is AAG. So there is a mistake here. G and U are not complement to each other. However, if you notice carefully, the G is actually complement to the C, which is next to the U. So what happened here? The whole ribosome shifted. Okay, the whole ribosome shifted one nucleotide forward, where this AA supposed to be AAA that complement to UUU. Now this AAG is now complement to UUC here. As you can see here, it moved one nucleotide forward. So now this becomes complement to the UUC of the original nucleotide. This is what it means by +1 ribosomal frame shift. Alright? It just moved one nucleotide forward. Alright? So what happened here? It's quite risky because the nucleotide is the evolutions of the virus has been millennia, and the nucleotide is very well conserved. And when this happens, you might come with a stop codon earlier than it's supposed to be, alright, or later than it should be. So in the end, what happened is we are producing a protein, PAX. We are producing a protein that is 20 amino acids lesser. So they share the same PAX and PA. They share the same N terminal, which is 191 amino acid, but it has a 41 amino acid that is truncated. So in total, there are 232 nucleotides, whereas PA1 have 252 amino acid. 232 amino acid, and PA has 252 amino acid. Alright? Okay. So that complete the total of 18 protein that being synthesized, being produced by using 8 segments of the influenza virus RNA. So we have 8 segments that produce full length of 8 mRNA, become 8 protein, as I highlighted in red here. Alright, these are the 8 that will be produced by using the full length. Then we have alternative splicing that produce 1, and then 2, 3, 4, 5. So segment 1, 7, 8 will produce another 5 mRNA. So in total we have 13. Then we also have alternative initiations, which PB1 will produce PB1F2, PB1N40, and then we also have PAN155 and PAN182. So here produce another additional of 4 protein, and last one is +1 ribosomal frame shift that produce another additional protein, which is PAX. So listed here are all the functions of the proteins that are being produced by the virus. and each of it has the important characteristic and important. Those that highlight in red are those that are really important for the replications of virus. So you have here induced cell death, host innate immunity regulation, and so on and so forth. So a lot of these proteins are still under investigations. Right? So now I would like to take a step backward. Alright? So remember we mentioned inside the nucleus, when the mRNA is being produced, the mRNA must be capped, must be spliced, must have poly-A tail before it is allowed to go out from the nucleus. So these are the three main things that is important for an mRNA to be exported out from the nucleus. However, we know that the full length of the protein by the virus are not spliced. They have cap, they have poly-A tail, but they are not spliced. When they are not spliced, how do they escape and go out from the nucleus? So apparently, this is a hypothesis, apparently the viral mRNA, alright, utilize the nuclear export factor NXF1 to allow them to go out from the nucleus. So it employs first protein, nuclear cap binding complex, CBC, that bind to the 5' cap of the mRNA. It also employs this CBC. Once it bound, it will then attract transcriptions export complex that subsequently attract the nuclear export factors to transport the mRNA out. They also employ the CpSF6 poly-A tail binding factors that binds to the poly-A tail of the mRNA and also recruit the NXF and export the mRNA. So we also found that the NS1, NS1 from the non-structural protein from the virus is also responsible on recruiting the NXF to send the mRNA, to transport the mRNA out from the nucleus. So this is the mechanism that explain why the unspliced mRNA of the virus can be exported out from the nucleus. Alright, so now mRNA is being produced, go into the cytoplasm, ribosome translate the protein. So in the cytoplasm, we have all this protein is being produced in the cytoplasm, and the endoplasmic reticulum (ER) will produce those that are required glycosylations such as HA, NA, and the M2, the ion channel. So these three will go through the ER, the Golgi apparatus, trans-Golgi network, and then display it on the surface of the host cell. Whereas the rest of this protein, they will be translated in the cytoplasm. And what happens next is some of this protein will be translocated back into the nucleus. So I specifically highlight those in red, indicating that these proteins are really important and it must be transported back into the nucleus. Alright? So why are these proteins so important? As you can see that NT protein is definitely the one that you want it to be in the nucleus, because the RNA is being produced, they need to encapsulate the RNA to protect the RNA. Alright? Then we know that the genome of the virus is panhandle structure, the panhandle structures. So it has the PA, PB1, and PB2 that bind to the nucleocapsid protein. So the rest of this protein are also play a very important role, which I will explain later. So basically, this highlighted red protein will be translocated or transported back into the nucleus. So just like NDV, where we know that the viral protein has been produced a lot, and then in order for the polymerase to stop synthesizing mRNA and start producing the viral genome, the NP is the one that's responsible for the conversions. So in viral genome, for influenza virus, the similar hypothesis is being proposed, where the NP is the one that once they go into the nucleus, alright, so they will now bind to the original ribonucleoprotein, the RNP. So we have PA, PB1 and PB2 that binds to the nucleocapsid. Now the NP will now bind to this ribonucleoprotein and indicate the polymerase to stop synthesizing mRNA. Instead, they start synthesizing the viral genome. So we have completed the third step, where the synthesis of viral protein. Now we come to genome replications. One NP binds to it. What happened next is now they use the dinucleotide, right? The dinucleotide, which is the, now we can see this is the 3' to 5' of the genome, right? This is where the poly-U located, right? So the two nucleotides that is important that serve as a primer for the replications, all right, of the virus is A and G. So what happened now is A and G dinucleotide inside the nucleus will now bind to U and C, all right, because they are complement to U and C. A, G serve as a primer, and now the polymerase will start synthesize from beginning all the way until the end to produce a positive strain RNA. Okay, viral complement RNA. This is a positive strain. Now, when this is being produced, ribonucleocapsid, the NP protein, will immediately bind to it. Okay, the PA, PB1, and PB2, the polymerase, will also bind to it. Alright, they will also bind to it. Alright, the next step is this will now serve as the template. The same process will now carry on. Alright, A and G dinucleotide will bind to here, and they now start synthesizing the 3' to 5' viral genome by using this positive strain viral genome as a template. So now in the nucleus, they will start synthesizing a lot of this viral genome. The same thing happened as the nucleotide started to come out. Immediately, the NP will bind to it. The PA, PB1, PB2 will bind to it and form a panhandle structures inside the nucleus. Alright, so now we've completed the fourth step, which is genome replications. So once the negative strain RNA is encapsulated by the NP protein, this protein or the ribonucleoprotein now need to be exported out from the nucleus. So NS2, remember I mentioned that the proteins that go into the nucleus, NS2, or another name for NS2 is nuclear export protein, NEP. Now NEP is a very important protein that it interacts with the host cell export protein, or CRM1. It interacts with host CRM and signal the CRM to mediate the export of RNP into the cytoplasm. Alright, so this is a very important protein that needs to be transported into the nucleus to allow the newly produced RNP or genome, alright, to be exported out from the nucleus into the cytoplasm. So what is the role of M1 here? Remember, when the virus infects the cell, the RNP contains a signal peptide, okay, or nuclear localization signal, alright, nuclear localization signal that make the RNP to be transported into the nucleus. The same thing happened to the newly synthesized RNP. They also have this nuclear localization signal. So if this signal is being recognized, we will be continuously going into the nucleus and it will never be able to bud out. So what happened now is the RNP will now be surrounded by the M1, the matrix protein, where the matrix protein will be the one that cover the RNP and prevent it from exposing the nuclear localization signal to the protein or the host cell transport system. So this prevents the re-import of the RNP. Alright, to transport the nuclear to transport the RNP back into the nucleus. So now we have the ribonuclear protein that's surrounded by matrix protein exported out into the cytoplasm. So what we need to do next, the virus need to do next, is assembly. So what happened here is what we can see, 8 segments is required for it to bind to the glycoprotein on the membrane. And as this happened, you can see the M protein started to dissociate and started to form a circular shape and getting ready for this virus to bud out from the host or from the cytoplasm. As we can see here, the virus started to bud out. Now bear in mind that in order for this to bud out, the virus required 8 segments, right? So studies have shown that if there is only 7 segments that come to the M, alright, or come to the newly synthesized glycoprotein area, it will not be assembled or bud out. Alright? There are certain mechanisms that to ensure there are 8 segments of the RNA to be together in order for it to start budding out from the host cell. So when it bud out, neuraminidase, the NA, will now play a role to remove the salicylic acid receptors from the host cell. This will prevent the virus from reinfecting the host cell. And now the newly synthesized virus will be released to infect a new host cell. So we have discussed the life cycle of influenza virus, where we talk about how it attaches, it fuse, and then viral protein is being produced. Now we have 18, so this is slightly more complicated than the NEV. So I hope you enjoy learning this. And then genome replications is quite similar to NEV, right? How where the negative strain produce a positive strain template, and then the positive strain template is used to synthesize more of the negative strain viral genome. Then assembly occur, and then budding or eclase. Now for the next slide, I would like to introduce you to 2 terms, because when you study influenza virus, basically these are the 2 terms that we always encounter: antigenic shift and antigenic drift in flu A. So we know that HA and NA is very important. They are displayed on the surface of the virus. They are important for the virus to attach and to release from the host cell. It's a major antigen on the surface antigen of the virus. Now what does it mean by antigenic drift? Do you know that every year that the virus will undergo mutations and these mutations that occur will not be recognized by your immune system, and when it is not recognized by immune system, they can start infecting. So that's the reason why you need to take new vaccination every year for flu. So the mutations that occur here is called antigenic drift. It's actually a selection of variant by the immune system. So remember the HA protein, you have one of the example is the HA protein that we have seen earlier on, where the HA is being cleaved into HA2 and HA1, and underneath I show you there are a lot of glycosylation site. Now I did mention that the glycosylation site is also one of the important factors that making a virus to be virulent or pathogenic. So antigenic drift, sometimes by changing the glycosylation site, for example, remove one of the glycosylation site or additional of one of the glycosylation site, that is also make the virus to be not recognized by your immune system. So this is what we call antigenic drift. But this process is relatively slow. For example, it might take a year for it to start developing another drift, so that your body cannot recognize it, and then it starts causing infections again. That's why we have seasonal flu every year, and it only resistant partially in the host cell. But the one that is more serious will be antigenic shift. Now, this antigenic shift is a result of recombination, or we call it reassortment, right? Now I'm sure you already know that the RNA have 8 segments of the RNA. So one of the segment, which is HA or NA, so there are a total of 16 and maybe now 18 or even more HA gene. So there are a total of 16 to 18 subtype of HA, which is being coded by the HA gene. Alright? So there are also total of 10 subtype of NA. So what happened here is, because they are segmented, meaning that the RNA can be sorted, means that they can take maybe from this virus HA combined with the other virus NA together, then they will start producing a new subtype. So the difference between this this subtype is about 30 or more amino acid sequence. So reassortment is the one that can cause pandemic. Alright, that can cause pandemic. So the thing with flu A, as I mentioned earlier, they are flu A can infect not only human, they can also infect birds and swine or pig. So most of the time, the reservoir for flu A is birds, the migratory birds, as I mentioned earlier. So the virus, influenza A virus, can infect the birds, but it doesn't cause any symptom. So the virus will go into the birds, and they can replicate, and then the bird shit everywhere. All right. So release all the virus into the environment of reservoir. All these migratory duck, normally they will station at area where there's a water reservoir. So that is where they spread the shit and started to spread the virus. All right. So unfortunately, the birds contain all the 16 subtypes and 10 subtypes. Now, the H1N1, what it means is that you have subtype 1 from HA combined with subtype 1 from NA. That's why it's called H1N1. So imagine there are a total of 16 HA and 10 NA. Imagine the combination. You can have HA1, H1, and N1 to N10, then H2, N1 to N10, and you can multiply. Right? So if there are 16 times 10, there will be 160 subtype of the flu A. Luckily, the virus that causes pandemic are very limited, and they are normally H1, H2, H3, and N1, N2, and so on. Alright? So I would like to show you the history of the influenza virus that caused epidemic or pandemic worldwide. I think the one that causes the significant pandemic from the human history will be the H1N1. Now, I would strongly urge you to go to CDC website and search for Spanish flu, and then you can learn more about how Spanish flu occur. And this is the period where World War I occur, and a lot of people stay closely, the army, the doctors, they stay closely together, and that's create the outbreak and spread worldwide. And you just look at the CDC website and search for H1N1 Spanish flu that occurred in 1980. That will tell you a lot of story. Alright, so I think this is the list of history for flu that causes epidemic worldwide. I think it will be easier for us to look at it in here. Now H1N1 that causes we call it Spanish flu. This is the one that caused pandemics that killed tens of millions of people worldwide. Alright, so study have shown that the one that causes such a significant why this particular subtype is so virulent is because of the HA and the NP protein. These are the two proteins that are responsible for making the virus really virulent. So when this Spanish flu gone, right? Bear in mind the virus doesn't begin in Spain, alright? It doesn't begin in Spain. So the reason why it gives the name of Spanish flu, you can read it from CDC, right? So remember we talked about antigenic shift. The dangerous part of antigenic shift is when you have a combinations of original. For example, in this case, we have H1N1 Spanish flu, and when there is a reassortment, here what we can see, NA, HA, and PB1 come from avian H2N2. When this avian H2N virus genome combined with the original H1N1, they now create a new viral subtype that is not recognized by your body. Okay, so let's say for example someone who has been exposed to H1N1, so the body will develop immunity against H1N1. However, due to the reassortment, and what happened here, they changed the surface antigen from H1N1 to H2N2, and this H2N2 comes from an avian H2N2. So now this new subtype go into human, your human body cannot recognize it as the original H1N1, even though your PB2, PA, and NP, M, and S is still the same, but because the NA and HA already changed, so the body cannot recognize them as the original virus. So this causes another pandemic or epidemic. We call it Asian flu in 1957. Then what happened is, normally when a virus circulates, they will eventually die off when a new strain of virus coming out. So what happened here, this evolved into another one. What happened here, there is a change of the HA as well as PB1 from another avian flu virus. So it takes the PB1 and HA from this particular strain, and now what we have is on the surface of the virus, we still have the same NA from the Asian flu, but we already have a different HA from a new avian flu. So again, this will not be recognized by your immune system. So in 1968, there is another flu called the Hong Kong flu. Alright? So the next pandemic that is more, that caused pandemic in worldwide is called the H1N1, right? Pandemic 2009. So what happened here is not only the HA and NA or other viral genomes that go into the new subtype, we also have human, swine, and avian that come all together, reassort all the RNA and produce a new strain of the H1N1 that has all the combinations of the existing influenza virus. So this is the one that caused pandemic in 2009. Again, if you want to study more about this, you can go to CDC website. So this shows how dangerous it is for flu virus because of these reassortments that allowed a new virus to be created. So you might wonder, virus shouldn't it be host specific, right? So remember the virus, the HA of the influenza virus, they infect the sialic acid receptor, which is present in human, avian, as well as swine. So I would like to introduce you to this sialic acid. So what we can see here is there are two types of sialic acid. One is alpha-2,3 and the other one is alpha-2,6. Over here, what we can see, when you have 2,6 means that the linkage between the sialic acid 2 linked with the 6 carbon here, whereas 2,3 is here. Alright, so this is 2 and 3, sialic acid number 2 and 3 from the galactose. Alright, in avian intestine, they are predominantly 2,3. So this is where the influenza virus can infect and go to the intestinal cell and replicate, and that's why I say when the migratory duck or bird shit, they will just release all the virus into the shit. Alright, whereas in human trachea, because it's airborne and in fact the respiratory system, we have predominantly 2,6. Alright, predominantly 2,6 linkage, this one. However, for pig or swine, their trachea have both 2,3 and 2,6. So this result in flu virus that only recognize 2,6 can also infect swine. Flu virus can infect 2,3, can also infect swine. So what happened here, the swine or the pig can now become a mixing bowl. So any virus that can infect birds, any virus that can infect human can also infect pig. So imagine in one cell where this particular cell has been infected by avian flu, then human flu go in, then the pig also infected by the swine flu. So in the end, inside this cytoplasm, what do you have? You have virus, 8 segments from avian, 8 segments from human, 8 segments from swine, all now become jumbled up. So I mentioned to you, in order for the virus to bud out, it needs 8 segments, but it does not specify the segment must be come from the original avian, come from original human, come from original swine. As long as they have 8 different segments, it will now budding out. That is the reason why the reassortment or antigenic shift will create a new virus that causes a pandemic worldwide. Alright, so that's the end for part 4 for enveloped negative strand RNA virus, especially for segmented RNA virus. That's it. Thank you.